great to be here. This is a very nice conference. Yeah, I'm from University of Bielefeld in Germany, and this is actually this paper which we produce is actually a collaboration between our bioinformatics department and our pure informatics department, which I'm actually part from. So if I'm a bit spotty on the biological details, please forgive me. But in general, yeah, I'm very happy to answer any questions. Let's start maybe with the motivation. So we wanted to generate some synthetic genotypes. Why do we even want to do this? I think most of you have worked with like private data in general, like in bioinformatics, this is a big topic. And actually there's a big problem in general with like getting access to your data, I think. Typically this can take up to a year or even longer. And I think most people had some of this pain that you have to wait for this. And of course, when we generate synthetic data, we can maybe circumvent some of this by being freely able to share this synthetic data, because of course it's not attributable to a single person, it's not subject to the same privacy rules. So this could be a solution to maybe iterate quicker on your research ideas. And why do we choose this whole genotype data? It's actually also another important question. We choose this whole genotype data because we had in the bioinformatics department, they actually worked previously on some diseases like ALS, which actually were very important that you took into account this whole genotype and not a single cell gene position, because it's not basically not attributable to that. But if you have a more complex classifier which takes into account the whole genotype, then you can actually identify this quite nicely as this disease. Okay. Yeah. Okay, I think that's mostly it. Now I will go a bit into the pipeline. What kind of data are we using? So I said whole genotypes. This is like 3 billion base pairs for a whole human genome. This is, of course, a bit much to generate at once, at least if you want to use a bit more complex methods like deep learning. So we have to pre-process typically in biology. I think this is done. So first, the coding regions of the genome are identified and split into genes, and then single nucleotide polymorphisms, these SNPs, are identified, and these are typically what is actually analyzed. So we took some of this kind of data. So we took this SNP data for humans. We actually did even more compression, because I think like a typical human genotype is also classified by a couple million SNPs. Still a bit too large for typical deep learning approaches. This work was compressed using PCA with actually very little loss of, very little error. So these SNPs are actually like very interdependent. So we can compress them quite nicely without much information loss. This is the type of data which we actually worked on, just to clarify this. Okay. Now I want to talk a bit about what are the possible generative models to perform this task of generating a synthetic genotype. And this is actually very different from the architecture. So this is like a bit more of a general question. So I think most of you have heard about variation autoencoders. I think the previous talk also mentioned it. But of course, there are a lot more models which are used to generate synthetic data. GANs were quite big a couple of years ago. Generative Markov models are still nice and can be used to model transitions, which is basically like how evolution works. So they're still used a lot in biology. But I would say state of the art by now is like diffusion models. I think I think most of you have used them, like just trying around, playing around on the internet and generating some nice images. Yeah, I think everybody has tried it and they are working quite well. The quality is quite good. And of course, LLMs, also autoregressive models in the LLM style, so always predicting the next token, the next word. This is another type of generative model, but it has some downsides. I will go into like why we choose what we did choose. So of course, the diffusion model, like the title already suggested. So first I will want to go over the ones which are basically a bit older, but not bad necessarily. So first, the variation autoencoder. The output quality of this is typically not state of the art, and as our task here was to produce very, very high quality synthetic genomes, this is not the one we choose. Also, it does not scale well when you have like the compute to have like a really, really big model. Variation autoencoders are known to be like kind of unstable once you scale them to be too big. Actually, GANs also have similar problems. They are very unstable, hard to train, and in general, like it's just have worse quality than diffusion models and generative Markov models. Actually, they are quite nice to model these transitions, but actually a lot of the times the data you have afterwards, so you have to start at some real genome, then you model the transition, how would the child look like, how would the next child look like, but then you still have privacy problems actually, because you can still identify like that part of this genome was from this original genome. And so we didn't want to use those. So we wanted to either use diffusion models or autoregressive models. We'll go over those very quickly. So autoregressive generation, as I said, LLM style, like ChatGPT, just predicts always the next token. But more maybe here, what most people maybe don't know is it can only take into account information from the left, basically. So it cannot look into both ways. Like in the genome, like the interdependencies of the genome, they are not dependent on the directionality of on how it is read, basically. At least not globally. So you can have like from gene 1 can have dependencies to gene 8 and gene 8 also to gene 1. So they can interact. And the autoregressive generator actually puts in this assumption that this is not possible. So, and also we have a non-fixed length generation, which is basically also not what we really want here. We have like a fixed set of SNP data which we want to generate. So diffusion models are very well suited because they basically provide both of this. So we did go with diffusion models. Just a bit more overview of our related work. So actually there has been quite a lot of work on trying to generate synthetic genotypes data, because I think we were not the first ones who had like this pain point. Oh, getting data is really takes a lot of time. So there was a lot of previous research which I've marked on here. There's also like research which uses autoregressive models, but I think the main point here is the genome length. This is like way shorter. You cannot really do this for like the whole human genotype, and it's not really suitable for SNPs, where the positioning is very important. And also what is very important is this conditioning parameter here. So what we actually also wanted was we want to generate genotypes which have a certain type of condition. For example, which have a certain type of disease, which is from a certain specific global region. If you want to be able to do this, diffusion models, they are basically baked in. That's this capability. This is quite nice. So that was basically like in this research as the selling point. We are using like the newest kind of model, diffusion models, and we generate full-length genomes with, which we can condition on basically anything we have training data for. Okay, now I want to go into the model architecture. Because diffusion models can be realized with a lot of different types of deep learning model architectures. They can be realized with a transformer, they can be realized with a CNN and MLP, and also you can you can think about a lot of different combinations of those also, and also even other architectures. What we tried basically all three of these. They have all of their upsides and downsides. Transformers, of course, are like all the rage right now. They don't have any spatial bias, which is kind of nice, but which also means they need to use a lot of training data. Also, you have to basically tell them, okay, you are at this position now, which is actually very important with SNP, because, like, of course, it matters a lot on which genome you have this mutation. CNNs, they also don't know which position they are, so they also need this positional encoding, but they have a local spatial bias, which is quite nice, which reduces the parameter count a lot. And MLP, it has a lot of parameters, but that also means it overfits a lot. But it's actually quite nice to have some general structure, because it considers the general structure. So we'll go into what we choose later. I will just give you a quick overview of how, like, one, for example, the MLP unit architecture looks, and the unit in general looks. So first you get in your whole data set, basically, which is your SNPs, your 18,000 down-projected SNPs. They're already down-projected to 18,000 times 8 dimensions. Then you pass them through this unit, so they basically get compressed more and more until you're in this embedding layer. In this embedding layer, you can also do some cool visualizations, and then you basically do this up projection once again. So you basically try to reconstruct here your original genome. Okay, let's go into the evaluation. Now I talked a bit about how the model architecture would look like, how the generation would happen. How can we evaluate that after we've generated some genomes, that they are actually close to real genomes? Of course, this is a very interesting question, because in general this is not very easily solvable actually. For images, we humans can actually easily tell, okay, this is a good quality image, this looks like somewhat realistic. Also in images there has been a lot of work on these general pre-trained image models. For SNPs actually it's surprisingly hard to find general pre-trained models. There are these models for proteins, but not for the whole human genome at once, basically, because this is typically has been considered as too long, this is too challenging. We only want to look at the single gene, has a single gene like correlation to a disease and not has like multiple genes maybe correlated together, and then you get some combinatorial explosion. I think, yeah, this is a bit problematic. Okay, but I will go a bit more into the evaluation here. First, we start with our data. We have this training data from IRS, for example, 12,000 genomes, genotypes. This is actually quite nice, human samples. We split them into train and validation data. With the train data, we train our generative model, which produces then our synthetic data. And then we actually have some classification model, which is always the same. And this classification model is then trained only on the synthetic data or only on the real data. And then we can actually evaluate it on this real validation data. And if our only synthetically trained model is good, basically, so that means the synthetic data was good, then this recovery rate should be close to 1. So basically, we recover almost original accuracy. Yeah, that is the evaluation metric, I think, which is the most important, actually. We also choose nearest neighbor accuracy, but this actually had some problems because we have like this very high dimensional input space, so actually didn't perform very well in general, I would say, this evaluation metric. So what we did, we tried out 4 different architectures. As I said, also combinations are possible, so that's what we did here. And we have these three different metrics: this recovery rate, nearest neighbor adversarial accuracy, and we have some a use case, I would say, for practitioner. Maybe this is not really a metric. Okay, let's first go into this recovery rate results. So on the top, this is a model type which actually produces these results. And actually, I think what is also important to denote, actually, like to train these models and get good results, a lot of hyperparameter tuning had to be done. There's a lot of architecture-specific choices. So actually, a lot of work went into those, and we only choose, of course, the best performing ones and displayed them here. And these are different classifier types. As I said, we only compare the same classifier to itself. So this is the recovery rate always compared to itself. But of course, when we generate some synthetic data, we wanted to work on different types of classifiers. So that's why we visualize this here. And also we have different types of data. We have this ALS data and this 1,000 genome data set. I think this is actually also quite popular. Yeah, and as you can see, like for our best performing models, MLP plus CNN, we actually get out recovery rates in the range of 90 to 95% for most classifiers, which is really, really good. So that means like when our classifier only sees the synthetic data, it still performs with 95% of a classifier which has seen all the real training data. So yeah, this is quite nice. So this is the most indicative metric we choose to evaluate to say like that our synthetic data is actually good. Now we have some more metrics. This nearest neighborhood adversarial accuracy, and this can basically so these numbers, they should be close to 0.5. That would be like optimal. So as you can see, like still our model here is like more often than the other models close to 0.5, but it's not perfect on this data. But I would also say this performance metric is maybe not the best in this case. I think what would be really nice, and maybe somebody else can give me feedback later, is if people have any ideas on how to have more biologically inspired evaluation metrics for this type of data. But now I want to go into like a last maybe motivation for this and also result for our data. So we actually have this our synthetic data now, which we will be able to freely distribute. And now if a practitioner has, for example, like another small amount of ALS data, and they are not able to access one of these large resources, which is, I think, the case very commonly, they can train their own classifier with only like 5% real data, which they maybe performed in some cohort study, like only like 50 or 100 samples. And then they will, so if they would only use this data, they would basically get like very low performance accuracy for these type of models. But if they combine it with our synthetic data, they can almost recover the full performance of if they would sample on like 100% of our data. So this is quite nice. We can see like our synthetic data really can help people when they use it. And this we show for this ALS classification, the 1 kg data. This is actually another task, which is which was like the region classification. So this is 1,000 genomes. This was split into like 26 different regions. So you can also try this classification task, which is maybe medically not as valuable, but it's still a nice proxy to real performance. Yeah, and we can see that it also works on this type of data. Yeah, so I just want to conclude, like our synthetic data, so what actually is, well, what you can see is it does not just copy real data, but it is close to the real data distribution. And this recovery rate really indicates very nicely that we have high fidelity of our synthetic data, at least for the task. But also it should translate to other tasks. It shouldn't be a problem, but it has to be shown, of course. What are the next steps? Next steps are we want to train on a bigger data set. At the moment, we have like 12,000 human samples, which is already quite a lot for genotype data. But like UK Biobank had 500,000, so this would be nice. And we're working on that. Multimodal data, at the moment, we focus on the genotype data. Multi-omics, I think, is quite popular in this conference in general. Would be nice to generate synthetic multi-omics data, so we can share it even more freely. Yeah, and of course, more evaluation, I think, is also a big point. At the moment, the evaluation is kind of limited to this recovery rate and some measures which are not really perfect. And once we have finished all of this and we're very certain this will work perfectly, or at least good enough, I think we can publish synthetic data sets. And I think then a lot of people would be very happy to at least use this for brainstorming and trying out some ideas before they have to go through all the regulatory hoops of getting real data. Okay, that's it already. Thanks for listening.
Fantastic. Thank you, Philip. Great work, and thanks for sticking to time. We have time for 1 or 2 questions. So let's go over there in the back.
Thanks for the interesting talk. I was wondering, I mean, if you're planning to release this data set, how can you guarantee that it's private? Because we know that, I mean, if you're using diffusion models to generate tabular synthetic data, and there's a lot of research showing that these are vulnerable to some privacy attacks, like membership inference attacks. So why is your approach safer than these?
That's actually in general a very good question. It is, so there's like this one type of diffusion, we can talk more about it later, which you can actually guarantee, which is private. So this is privately guided diffusion. But then typically you have a quite large loss in generation quality of the genomes. So we tried this out and it wasn't perfect. So in general, like guarantees are always hard to get, I think, especially with deep learning. But what we can show is that at least in our distributions are looking close, and when we try to generate data, we don't get really, really close. So we don't have like this, we never observed this problem which was reported in other papers with diffusion models that they get exactly like basically a copy out of the training data set. We didn't have this. But at the moment we cannot guarantee it actually. So this is definitely some future work which also needs to be done. Yeah.
Right. One final question over there, the lady there.
Thanks for the great talk. Actually, we're also working on methodologies for generating synthetic data, so I'd love to have a chat with you afterwards. Yeah. I was also curious, following on from the previous question around the privacy aspects, is it able to sort of tune the parameters to sort of increase or decrease the level of privacy in the output data? So obviously there's a trade-off between privacy and accuracy. The more private it is, you're going to reduce your accuracy. But the less private it is. Are you able to control that in this methodology?
In this implementation, we actually didn't do this. The published implementation is actually another type of diffusion, like I already mentioned, which is actually able, I think it's called epsilon type diffusion. I have to look it up, which is actually able to actually perform this trade-off. I don't think it's so difficult to implement this, but we didn't, we actually tried it out. But in our experiments, the generated results were a lot worse, not only a bit worse. So yeah, this trade-off is quite large there. And also, yeah, at least in our we generated like over 10,000 genomes, and for us, these attacks didn't work on our model. So we tried them out a bit. Some of the described attacks in the paper But I think there are like so many attacks out there, so we cannot say for certain that none of these will apply to us. It's very difficult to make this provable. So yeah.
Okay, fantastic. Let's thank Philip again.